Our 10th finalist. From Fairport, New York, please welcome Bill Rader and Spencer Rosero from Raland Therapeutics. Thanks. You're going to do it all? Yep. You need help? Here's your,
yep.
There you go.
Good afternoon. I'm Bill Rader, the Chief Executive Officer of Raland Therapeutics, and I'm here today with Spencer Rosero, our Chief Medical Officer and the inventor of our core technology. Spencer is also a professor at the University of Rochester and a medical doctor. We're here today to talk to you about cancer, and more specifically, chemotherapy treatment within cancer. Now, I'm willing to bet everybody here has been touched in one way or another by this horrible disease. I know I have. I've lost my friends, family members, my mentor in life. Even both of my parents are cancer survivors. In fact, every minute of every day, 3 people in the United States are diagnosed with cancer. 1.6 million people per year in the United States alone. Now, 1/3 of all hospitalizations associated with cancer are treatment-related. We're here to talk to you about a problem in this, and that's chemotherapy dosing, okay? Chemotherapy today is dosed based on patients' height, weight, questionnaires, blood draws, many other factors. Now, oftentimes this dosage is too large, and this is toxic. This causes a lot of the toxic side effects and can really cause the treatment to be abruptly stopped because of blood issues. Now, if the dosage is too low, well, it's oftentimes ineffective. So you're really looking to be in this kind of just right area for the treatment. Now, what if we could minimize the delays in adjusting treatment, provide better and more physiological data to the caregiver so that they can better observe, modulate, and control the therapy? What if we could modulate the chemotherapy so we can eliminate the breaks in treatment because we're giving such a personalized dose of chemotherapy? Well, we have Cytocom. Now, Cytocom is an implantable biosensor that's very revolutionary and unique. It provides proactive rather than reactive treatment and provides more data to the caregivers so they can monitor you 24 hours a day, 7 days a week during treatment. This sensor uses living cells, the patient's own living cells, to act as the instrument to provide this information back to the doctor. We can detect, our value propositions basically, we can detect symptoms prior to their occurrence. So if we can detect that your blood chemistry is such that you're going to be feeling bad, we're able to modulate the dosage and prevent possibly some of this, some of the symptoms associated with the chemotherapy. Basically, we're getting more data, and we're able to utilize this data to provide better treatment in real time. Now, the sensor is revolutionary. This is patent-protected technology. We have multiple patents issued that uses the living cells, the patient's own living cells, in the biochamber, okay? We also have some telemetric data, or telemetric sensors in the sensor itself to provide us some additional data. Now, the software side of this is how we get this data from the patients. We've executed an NDA with the largest wireless carrier in the United States to help us with this, to help us keep this HIPAA-compliant data secure to the doctor. This provides the doctor, the caregivers, more data in real time using a secure network. It's also a revenue stream for us, the SaaS side. Now, in phase 1, we have to go through the animal market. These are the millions of animals that are used each year, the mice and what have you, that are used for drug development and testing. Here we provide the researchers real-time relevant data, and we use substantially fewer animals. Currently, they use individual animals are just a single data point. The way we do it, we can get multiple data points of a single animal, therefore using a lot fewer. This is a large market. There's over $4 billion spent each year on the lab rats alone, millions more on other animals. Here, we're going to hit the ARO, which is the academic research market, and clinical research markets in about 18 to 24 months with our first generation product. Okay, this product is very important, yet it doesn't require FDA approval to get there. Next up is the most personal to me and the most important. That's the cancer chemotherapy. Now, here we're going to be talking to physicians and hospitals and insurance companies because we're able to provide better treatment for less cost. We provide real-time, relevant data and allow the patient to have less suffering during this treatment. Now, this is a huge market. $35.8 billion in the United States alone is spent treating people with cancer. We're going to help to lower some of this cost by reducing the amount of negative effects on the patients. Here we're going to be in early testing in humans using stem cells in 2017. We'll be in full clinical use by 2021. Now, I've been able to assemble an incredible team of people. I did this early. I wanted to ensure our success. So I was able to recruit one of the top CEOs in the United States, a 4-time CEO actually, a top chief marketing officer who took pictometry from a $10,000 startup to a $650 million exit, a top researcher, and one of the top CFOs in the United States, along with Dr. Rosario and myself and others that have worked on this device up to this date. We also have partnerships with universities, and we also have advisors from the FDA. Now, we have 2 issued patents. We have additional patents that are in progress right now, and about 6 other patents we anticipate filing in the coming months. When it comes to competition, status quo is probably the biggest competition, what doctors already do. Now, there are other sensors on the market, but they're basically blood analyzers, and they don't use living cells. We're using the patient's own living cells to actually monitor the patient's condition, and this is huge. Now, to date, myself and others have invested pretty heavily in this. We believe in this company and its products. We've also been able to obtain a government grant. We've taken this money and we've put it to good use. We've designed and manufactured prototypes. We've done cell study tests. We've completed economic studies. We've done customer marketing and are currently in the labs doing follow-on cell studies. Our next steps, well, right here from Buffalo, we're going to be hiring people. We're going to bring some additional science, researchers, and even some IT jobs in. And that will help us to get to our revenue point in a couple years. Our financial projections put us at about break even by about 2017, and this is using early ARO and CRO cells. By 2018, we're profitable and we start generating revenue that'll help us to self-fund a lot of our continued research into humans so that by 2021, we're able to provide this product to humans. Now, in the last 10 minutes, there's been 30 people, just since we've been sitting here, 30 people diagnosed with cancer. 18 of these people are going to start chemotherapy treatment in the coming weeks. Join us in helping these people. We can do a lot. We have the team, we have the technology, we have the know-how, and more than anything, we have the passion. We want to help these people, and we're going to do it right here in Buffalo. Thank you very much. Now, I want to bring Spencer up. Thank you. I want to bring Spencer to the stage.
Spencer, you going to come out?
Come on.
Come on up here. David, obviously, first question. Comes to cancer. You're our resident expert. Gives everybody else a chance to think of their questions. All yours.
Very, very interesting work, and I share your enthusiasm for its potential. I think perhaps one of my key questions revolve around two areas. First of all, will be the sustained viability of your cells. And I think you're up to 31 days from a much lower number. That may not be enough for more longitudinal kinds of therapy. And I presume if cells get natural die-off, then you're going to have fluorescence conversion from their senescence rather than from the drug effect. Am I right or wrong about that?
So that's actually some of the concerns that we had early on.
Yeah.
And one of the issues comes as a couple of things, is cell survival itself. So initially, we only had 2 or 3 days, a lot of trial and error, a lot of the biomembranes. We were sampling from all over the country, different kinds of biomembranes, biofouling, some of the issues with implant stage. And a lot of it was trying different dimensions, where initially it was too big, too little, how much oxygen gets across, nutrients. And we found kind of a sweet spot in the design that can mechanically protect the cells, because they have to love the environment. At the same time, provide the diffusion capacity for the signals that we want to go across and for the cells. So we got to 31 days, and that's just for the pretty much a very tight string budget, equipment from eBay, a lot of things that we ended up piecing together. So we were able to do the experiments up to 31 days, and as a fact, about 3 years ago, we had a multi-billion-dollar company said, hey, if you can get it to 7 days, that'd be a big deal. So for us, 31 days is a big deal. Chemo, we're thinking right now it's 31 days, but that's without running full optimization studies, without changing the environment, without trying a host of other biomaterials, because the first point was get them to survive, get them to function normally. And we are looking at ratiometric as a capability for the cells themselves, for fluorescence, for response, but also for viability using some of the newer techniques in optics that weren't around 8 years ago that could be miniaturized like FRET.
Because I think many chemotherapy regimens will be 6 weeks to 3 months long. And is there a way to recharge these things?
I think one of the ways we've spent a lot of effort in the beginning to miniaturize a bit, because it may be as simple as a small injection. So the device with miniaturization would be the injection. The cells live in their environment, but the cells are personalized. It could be for different applications. But they can be put in and it re-injected at 6 weeks or 8 weeks.
Okay.
So compared to the overall toxicity with hospitalization and everything, its risk-benefit is probably in favor if you have a good monitoring system that can
And there
keep
is
you
a
out.
percutaneous re-injection port so that you can go through the skin to...
No, one of the things is a lot of advances have been made. I do electrophysiology and implantable loop monitors. And some of the most recent ones are 3 years of power supply with an injectable format that could be slipped out at the end. So we may have to, I think at the beginning, the reality is it'll probably be 1 or 2 injections over a 3-month period, and then you remove the device at the end.
All right, good. My other question was around the specificity of the cells relative to the drug that might be used. Will you have to tailor the cell type to particular mechanistics of the drug?
That's actually the beauty of the thing, is that the cells are the sensors. The electronics, the housing, all that stuff that we can fabricate and manufacture, that is the platform that holds the cells in their environment. So if we're looking at bone marrow, red blood cells and white blood cell toxicity, which is neutropenia, which is very common in chemotherapy, is we can start looking at the precursors, like the stem cells or the precursors or cells that are the most susceptible to the chemo. So if we calibrate the cells, it's no different than other sensors, you calibrate them, so when they go in, you know that, hey, can I pick up a change, a protein, a signal 20% earlier in time before the person starts to get sick? And then that's the only signal you need to say, hey, let's keep giving the drug, but now we can back off on the dose, but not have to stop after symptoms develop.
And lastly, how big is the box?
Well, right now, the microenvironment, which is actually, that was the hardest, is right here.
Oh, okay.
He's holding it. I can actually bring it over. Are we allowed to do that?
Sure. I'm dying to see it.
So,
others too,
stuff
maybe.
from the front? Or?
Kim, you have a look.
Yeah. So it looks very simple, even though a lot of work went into that. One has an optical window on one side. The other side has the biomembrane. And we control through the biomembrane the pore sizes and what can go in and what can go out. And then, so the cells are happy. Now, that cartridge is the sensor. it slips into an optical unit, which we still have to miniaturize. We did not spend a lot of the precious money early on to optimize. And for the optics, because electronics, the hardware, all that could be done. The biology was the hardest, so it was to make the friendliest environment we can for those cells, and then the optics and the hardware come later, because there's no technical hurdles in that right now.
Yeah, we currently have a desk-mounted reader that we use.
Okay.
So that's how it's working now, and that's how it'll be working in the version 1 into animals.
Does the reader control an infusion pump so that you get continuous regulation?
It will in the future. Right now, what we do is, since it's only 50 microliters, small amounts that we put in it to measure, it's still on a prototype stage. Infusion, monitoring real-time feedback into infusion systems, or just an app that just helps them gauge the therapy, because right now we do everything body surface area.
Thanks, and I've talked too much.
Okay, sorry.
Esther.
So this is less about your specific product and more about what you hope to find out. How much, there's dosage in general, and then there's things like timing. Do you take it in the morning or the evening, after eating? And I have a suspicion that you can get huge benefits there as well. I'm just wondering what the science is around that.
So it's, when trying to take quantitative measurements in biology, it depends on the protein of interest or the signal of interest. So some signals are seconds, maybe a few minutes to peak, and then they die off, they wash out. Some other signals, for example, in thyroid disease, where the hormones, it can take days and weeks to cycle through. But all we have to do is the cells do their own thing. They don't know they're in a chamber. They're just they're sensing, they're responding to the signal. The hardware, if we know it's a protein that we know can go up and down 3 times a day, or a signal, we can tell the hardware, probe, there's another word for it, I can't remember right now, or ping.
Yeah.
Turn on the light, get the sensor data, do 50 samples, get a statistical analysis, and that's it. And so the hardware can be programmed to say, I want you to, this cell line, I only want you to check it once every other day, or once every minute.
That wasn't really my question.
Oh, sorry.
Though it was an interesting answer.
I misunderstood.
The question is, if you take the same dose, but you do it twice a day versus once a day, if you give it in the morning versus the evening, there may be a way to lower doses even more and get better impacts, less toxicity, depending on the timing, their circadian rhythm.
So the area under the curve in the clinical trials, how they decide dosing, is to maximize the efficiency while reducing toxicity. And actually, there's an expert right here among the judges for how chemotherapy is. But we could go back, and I have not done it for a particular chemotherapy, but we could go back and figure, hey, instead of the twice-a-day bolus dosing, which is a standard, you get a peak and you get a drop, can we give it more frequently and reduce toxicity? Or can we give the initial bolus and then figure out what the next dose should be? But we haven't gotten that far yet, because the first hurdles were just keeping the cells alive in the implanted state. And
Thanks.
Bijan.
Is there any kind of geographic or demographic variance on overdosing? Like, is there, like, or is it, do we have an industry issue due to a technical limitation, or is there expertise that is developed in some areas and not other areas? Like, what's the gravity of overdosing right now?
So I wouldn't use the word overdosing per se, even though that can happen. If it happens, it would be like an accidental overdose or a miscalculation. I would say that chemotherapy and a lot of other treatments we give all have side effects. Most drugs have side effects. It could be even Tylenol at high doses. So the question is, how can we minimize the toxicity side effects? And for chemotherapy, the side effects could be pretty significant. They may result in hospitalization, and quality of life. So I cannot say at this point, this is going to save lives. What we can say is that the more biological information we have, the more we can personalize the dose, and then we can improve that ratio of benefit versus risk and benefit versus toxicity. So right now, if chemo had no side effects at all, then it probably wouldn't be this would be primarily a research tool. But for clinical use with side effects, chemotherapy short term especially, it looks like it's the ideal application to help with symptoms.
Well, maybe just quick follow-up if I might, and this is a naive thing, but the first slide just gave me this feeling that we have a dosage, side effect relationship, right? And I'm trying to figure out, is that state getting better, worse, staying the same? You know, like
I believe over the last 50 years, 60 years, the chemotherapy is smarter. It's targeting statistically. You grab the different people, and then you say, you benefit more from this chemotherapy. You benefit, another person benefits from a different chemotherapy. So the state of the art is getting better. The way we give it, we still want, for the oncologists, they still want the maximum killing of the tumor, but spare the good cells. But the tumors were originally good cells. So the good cells still get hurt along the way. And it's always that ratio, and it's a tight window because you don't want to give too little, and you don't want to overshoot as well.
Okay, thanks.
Last question.
I have a quick question. How much money do you need to raise in order to bring it to market, and over what time period?
Okay, we need about, currently, to get us to our next step.
No, think all the way out.
Oh, all the way out. It'll probably be somewhere around $30 to $50 million.
And that's, what year was that?
That'd be that would be in the total time between basically 2017 to 2021. Okay. And we'll be generating a lot of that revenue in our early cells in our preclinical market. So will we have to raise all that money? Possibly not, but we may need to raise a part of that at least.
Okay, thanks.
Thank you very much.
Okay, thank you very much.
Thank you.
Doc, thank you very much.